Okay, so thanks Satoshi for chair, and I'd like to give my thanks to Miu and Christian and Klaus for organizing this wonderful meeting. And also I'd like to give the previous two talks from Miu and Satoshi's lab, not only give background, but also give some common studies that make my talk easier. So our study focused on the gravity sensing in plants. And this is what we took in the dark condition, use far-red light. You can see that when the seeds germination, the roots and shoots growing toward all different kind of directions, but they will recorrect to the downstream growth and the upstream growth. So this is essential for the survival of the plants. I just want to use this photo to emphasize the significance of gravitropism. So for how plants sense the gravity, actually there are three scientists, Nimek Habland and Francis Darwin. He's the son of the Charles Darwin. They proposed the statolith hypothesis 120 years ago. So they proposed that the sedimentation of aminoplast in the epidermis cell and the columella cells, they can perceive that plant is no longer in the vertical. So this proposed to start the gravity sensing, but there's a long last question, as Miu and several others talked, how the physical information of the aminoplast sedimentation to be converted into physiological signal. And this is a long last question in this field. And also another star gene we already talked a lot is the lazy mutants. Actually, this mutants was identified, also have a long history, kind of in 1931 was first identified in the maize, and then in 1938, it's in the rice. This kind of mutants was identified. But the genes clone was until, as Miu introduced, is until 27, and also a Chinese group and a Japanese group identifying the lazy one gene, the founder member of this lazy family, first in the rice, and then other studies in the maize and the Arabidopsis. And also in the roots recently, several groups found that multiple lazy genes was involved in the root, but the phenotype a little bit different from different groups. So some of the groups show that the mutation of these three lazy genes, they show opposite growth of the root. But some others show when knock out these three genes, almost lost the gravitropical response. There are small difference there. But anyway, our tell that the lazy genes are important for the gravity signaling. I paid attention to the lazy genes because when I studied the light signal together with Professor Tsinghua Deng, that's another reason I'm very glad to join this meeting to meet several photobiology scientists. So we found that when in the dark, the Arabidopsis show very nice negative gravitropical response. But in the light, especially in the red light, they inhibit the negative gravitropism. And also, but for the root, it's kind of promote the growth. So this can be explained somehow by the aminoplast development. But also we found another reason for this is the lazy genes expression. So in the hypocortical, the PIF genes can directly binding and regulate the lazy force gene. When during the dark to light transition in the hypocortical, the PIF degraded and the lazy gene dramatically reduced. But in the root, it's an opposite situation. You will find that the Hy5, another transcriptional factor accumulating in the light, they will bind the lazy and increase the lazy expression. So this, we can use the lazy expression to explain how light regulates the two organs of the Arabidopsis. And then we found that at that time, the lazy genes were still poor to be understood. We used the CRISPR to knock out all the three genes expressed in the roots. And in our hand, in the Columbia background, knock out all these three genes, we show the random growth, the growing up, down, side, all kind of directions. This is a statistical result. And you put either one of the genes into the mutant background, it will rescue the mutant phenotype. Okay, then we check how these lazy genes regulate the expression of the auxin genes. when you change the direction of the root, the auxin accumulates on the lower side much more compared to the upside. So in our lazy triple mutant, you will see that the asymmetric distribution of auxin all disappeared. So our expression level pattern was consistent with the phenotype very well. So that's we concluded that the lazy control graph tropism via mediating the polar auxin distribution. And then we fused the GFP with these lazy genes and get the localization. All these lazy 2, 3, 4 genes localize very well in the columella cells, and the lazy 4 with some epidermal expression. And in these cells, look closely, you can see the aminoplast localization and also in the plasma membrane, and with always on the lower half of the membrane. This is consistent with Mills result very well. So we found that there are these two localizations. One aminoplast is local, is connected to gravity sensing, and also the direction on the membrane they will show and probably control the direction. And we further use the immunolabeling to confirm that actually they localize on the plasma membrane and it's binding the phospholipid on the membrane, make the localization possible. So we continue to ask, are there any relationship between two localizations, aminoplast and the plasma membrane? And also in this part, we did two kind of rotations, it's 9 degree and also 180. You can just look at this for simple. At the beginning, the aminoplast on the lower side and the polarity on the plasma membrane. When you reverse it, the time goes, the aminoplast green, the drop down because of the density. And then the lazy protein relocalize on the lower side. And even during the process, when the aminoplast was adjusted to some membranes, and there they accumulate the protein. So it means that the position of the aminoplast with the plasma membranes accumulation is very well correlated. And then we want to kill the aminoplast to see whether the redistribution will disappear, but we could not. And then we also choose the starchless mutant. So in this mutant, the sedimentation was dramatically reduced, and then you can see the redistribution was much slower. And also we did a similar, the translocation test. We use the FRAP technology. We photobleach the two adjust columella cells plasma membrane signal. And for the cell 1, we keep the aminoplast. For cell 2, we kill all the signals. And then we check the fluorescence recovery. You can see that in the cell 1, the membrane signal recover quickly, but not cell 2. So it means that there are some protein can translocate quickly from the aminoplast to plasma membrane. So for this cell biology, I'm very glad that our results consistent with Mills and later with Yuri labs result very well. So sometimes the cell biology result, there's some confusing, but we all get to the consistent result. I'm very glad about that. And then we further ask, how does the gravity stimulation mediate the activity of lazy proteins? So we just grow in the Arabidopsis and then rotate it. During the different process, we do protein mix analysis. And then we found that the lazy proteins at the very beginning or after 24 hours, they show very low phosphorylation. But during the process, even after 5 minutes, very quickly, they show a dramatic phosphorylation increase. We further identify which kinase contribute to this modification. And we found that at least 2 kinases in MAP kinase pathway, MAP kinase 3 and MKK5, at the vertical, they show no interaction. And after 20 hours, there's no interaction. But during the process, the gravity stimulation process, they show dramatically interaction increase. And also our in vitro experiment can confirm this result. When you put these 2 kinases together, can interact with the lazy. And either one, it doesn't work. You need to put these 2 together and then do in vitro phosphorylation. The size are consistent with in vivo ones. That's we further check whether this phosphorylation size really have physiological significance. So we mutate these phosphorylation sites and then we put them back into the mutant background. So they could not rescue the plant phenotype very well. We check the localization. So you can see that in the white type, lazy 4, you can very clear localization on the aminoplast, these dots inside of the cell. But the mutants are most clear within the cell. There's no clear aminoplast localization. And also for the polarity, after gravity stimulation, the lazy accumulate on the lower side much more than the upside. But for this mutant, the asymmetric distribution was dramatically disrupted. So it means that this phosphorylation are important for lazy localization on the aminoplast and for the polarity. So we reduce the two MAP kinase and result in similar results. Then we further ask, how does lazy localize onto the surface of aminoplast? So as Neil mentioned that the Patrick Masson's group, I did a very nice screen. See, this IRG mutant, the phenotype is very weak. You can almost not find it. You do graph stimulation, you can find some phenotype. But they did an enhancing screen. They found this MAR1 and MAR2. These are TOC proteins. TOC means the translocan at the envelope of the chloroplast or at the aminoplast. And then you can see this random phenotype. But the single mutant of TOC is also weak. So this mutant was identified for many years, but doesn't understand how TOC works in gravitropism. To look at these TOCs, there are several TOC complexes on the outer membrane of the aminoplast. Also, we found that the lazy can accumulate on the surface of aminoplast. Then we just think about whether these two proteins interact with each other. Then I ask students to do the interaction test, to test the lazy with all the TOC proteins. But during 2 years, we could not detect the interactions. We think about what happened until we found that lazy can be phosphorylated after gravity stimulation. We use the mimic lazy to check the interaction with TOC proteins. We found the lazy can interact with TOC 34, and here is 120 and 133. And all these 3 TOCs, they exactly form the same TOC complex, this one, but not interact with all other TOC complex, and not interact with the TOC 75 within the membrane. So it means that the lazy interact with TOC proteins components facing to the cytosol, not go inside the aminoplast via the TOC complex. And we can in vivo the colocalization show very nice merged signal. Then because previously the TOC mutant show very weak the gravitropic phenotype, then we ask whether they are really physiologically important. Then we knock out the 2 TOCs, 120 and 132. Then we check the localization of lazy 4. We found that there's obvious defect in this mutant. In the white type, you can see that the lazy can accumulate on the aminoplast very well and always on the lower side. But in this mutant background, the lazy cannot accumulate on the aminoplast again. And also the polarity was dramatically reduced. Then we check the phenotype. You can see that during gravity stimulation, after 12 hours, the white type show very nice bending, but in the TOC mutant background, they grew toward the original direction, show obviously gravitropic defects. So take all this information together for plant how to sense in the gravity. So now we have this kind of model. So this aminoplast accumulate in the columella cells. And then they, in the regular growing condition, they sediment on the bottom. And with lazy accumulate on the plasma membrane. And when you do the reorientation, you will find that at the beginning, the aminoplast on the side, the MAP kinase, there's no interaction with lazy proteins. And the gravity stimulation will trigger the interaction between MAP kinase and the phosphorylate lazy. And the lazy enhance the interaction with the TOC proteins on the surface of aminoplast. Then during the sedimentation of aminoplast, will bring the lazy proteins to the bottom of to the new bottoms and transfer to the downstream oxygen signals to trigger the bending. Okay. Okay, so we put all this information together, we think we get to this kind of gravity sensing model to explain the aminoplast sedimentation theory. And the key is that the aminoplast can bring the lazy to the bottom. That's similar conclusion as Miu and Yuji. And we found that the phosphorylation is important for the lazy to interact with TOC complex to be recruited on the surface of aminoplast. Okay, then we continue to study these lazy proteins and gravity sensing in other species as septosis students just proposed and talked in a nice introduction about the phascometrium and also phascometrella. So this kind of the filament is quite different compared to Arabidopsis. Arabidopsis is a multiple cell sensing and response system, sensing in columella cell, and they transduce signal to epidermis to trigger the bending. It's a kind of sensing and bending in different area. But in this phascometrium, you can see that all the sensing and the tropism response is within the same cell. So we consider this to be a different sensing systems. And I'd like to take this chance to specifically give my thanks to Professor Mitsuyasu Hasebi. I started to use phascometrium to do the studies about 10 years ago, and I got a lot of plasmid and support from Professor Mitsuyasu Hasebi. Thank you very much. And first, we ask whether the plasmid sedimentation is also important because we cannot detect clearly the starch accumulation. So we just generally call the plastid. So in the plastid, after gravity stimulation for 1 hour, you can see that there's no obvious sedimentation or almost evenly distributed. But for some of the filament, you can find in a specific region, they can distribute to one side. Then you can find some slow movement if there's any. So we are still not sure whether the plastid sedimentation also involved in gravitropic response of the proteinema. As also the 5 lazy genes. So we checked and knock out these lazy genes. So in our hand, we knock out one of the lazy genes. You can see that the Y type, the proteinema growing upside, but in this mutant background, they're growing toward all kind of side. It's kind of random growth. So we think this lazy is essential for the gravitropic response. And also we mutate all the 5 genes together, resulting in similar phenotype. So we think that in the phascometrium, there's one key lazy gene. And we use this gene can rescue the Arabidopsis triple mutant very well. So this is the Arabidopsis triple mutant, the roots growing toward all different kind of conditions. This is the Arabidopsis the lazy can rescue. But we use the phascometrium, this lazy 5 put in the mutant background can rescue the roots growing direction very well. So it means that the very early lazy genes already have the function and can also work in seed plant. Surprisingly, this not yet. So this is also, we check this phascometrium lazy 5 genes in Arabidopsis, just the similar localization in membrane and aminoplast. This is different lines, a little bit different situations, but the general conclusion is that also localized on aminoplast and plasma membrane. This is a surprising result. We think their biochemical activity of the lazy should be the same in the mouse and Arabidopsis. But when we put the Arabidopsis lazies into the phascometrium mutant, they could not rescue the phenotype. This is the mutant growing toward different conditions, and this lazy 1 and the lazy 3 put back, there's no rescue phenotype. So this suggests that probably during the evolution, some other components in these 2 systems already changed. Okay, then we go further to check the localization of these lazy genes. This is the Y-type phascometrium. There's some autofluorescence signal. This is our transgenic line. You can see clear plasma membrane localization compared to the control. And also there are some dots. So at the beginning, we think, okay, probably this is the similar situation, also plasma membrane and plastid. But we found that this kind of dots are a little smaller compared to regular plastids. And then we use the mitochondrial trackers to check and found that this does colocalization with the mito tracker very well, this analysis. So this is a very surprise. So the Phascometrella is lazy localized on the plasma membrane, that's so fine. But I cannot say it localized on the mitochondria. But we can say now at least we have data to show they are co-localized with the mito tracker. And also similarly, we did not find obvious plastid localization similar to tissue labs result. So that's kind of confusing how will lazy work in this system. So a clear phenotype, different localization. Okay, so we don't know how it works. Then we think probably genetic screening in Phascometrium can help us to identify more mutants losing the gravitropism. Then we do forward genetics screen. This is the Y type growing upwards, and then we screen and look for those mutants growing randomly. And now we obtain the multiple, this kind of mutants, and not yet, the gene have not yet been identified. But very interestingly, we are also identifying some genes after the mutated these genes, they will show opposite growth phenotype. The proteinema growing upwards, and in this mutant, they're growing down. This is not involved in gravity sensing because if involved in gravity sensing loss, they will show random phenotype, right? So this probably means the signal transduction. But very interesting, I will show some of the results. So the first one is the gravitropism C. Actually, this kind of phenotype was first identified by David Cove in University of Leeds. Stephen is here. I visited there 5 years ago. Very nice to meet you again. Yeah, so this is a GTRC mutant is kinesin. It moved in on the microtubule toward the minus end of the microtubule. We found that is important for the direction to regulate the actins distribution, but not yet understand why. And then we continue to do screen. We identify another mutant, also shows similar phenotype going down. There's no functional domain of this protein, but also localized on the microtubule. Then we checked and found that these two proteins actually interact with each other. So this one should be this kinesin's adapter protein. And then also we get another one to growing downstreams. And this protein group, gravitropism E, localized on the plasma membrane. This is the unspecific autofluorescence signal. So for the membrane, it's a signal with some dots. During the gravity stimulation, they accumulate more on the lower side compared to upside, show some polarity. And very, very interestingly, this is the MPH3 is the homolog. So when you check this mutant phenotype in the phototropism condition, this one also show opposite growing directions during the phototropism study. So it means that probably these genes in the early land plant, they're growing both gravitropism and phototropism. And then in the seed plant, they divide it into the MPH3 and the MPY, different subfamilies to control the phototropism and the gravitropism. Okay, so summary, the result for the Phascometrium, we found that very interesting, there's no obvious sedimentation of plasmid in proteinema, if there's any, and no obvious plastid in rhizoid. Even there, you cannot detect clear plasmid there. And the lazy is essential in this system. And lazy is the localization, we probably on mitochondrial, but not plasmid, but we totally cannot make a confirmed conclusion at the current stage. And also recently we paid attention and just start to set up the system about cancer. The reason is that there's gravitropic response over the rhizoid, but there's no obvious plastic there, and also there's no lazy there. So indicate another type of gravity sensing system. So we'd like to use this system to trying to reveal the unknown gravity sensing mechanisms. And also probably in Arabidopsis, we also found some other, the kind of phenomenon cannot be explained simply all by the aminoplast segmentation. Probably also there's other revealed sensing mechanism in Arabidopsis. Okay, so I will stop here and give thanks to my group members at the Tsinghua University. And also I give my thanks to my previous supervisor, Xinwan Deng, and long time collaboration, and Dongjuan in Rutgers University. And my early study about the mouse phenotype is the mutants. Some of them are from David Cove and collaborated with Angel from University of Leeds. Okay, thank you very much. I'm happy to take questions.
So question.
Thank you for this very clear talk. In your model, where you implicated the MPK3 in the phosphorylation of the lacy, why do you put MPK3 downstream from the gravitropic signal rather than MPK3 being constitutively phosphorylating lacy so that lacy can react to the change in gravity?
Very nice and important question. So that's one of the questions we are trying to address. So we can use this model. You want to look at this? Okay, maybe I show the biochemical data. That's easier, right? Okay, so in the vertical growing stage, the status, we could not detect clear interaction between the lazy and the MAP23. Only during the gravity stimulation, the interaction dramatically increased. And so after they're growing downwards again, there's no clear interaction. So it means that this is the protein's abundance. We do the mass. We use the lazy for GLP to do as a bait, and then to pull down the proteins, we do the mass. And this is the abundance of these proteins in the mass. So this kind of dramatically interaction increase. So it means that the interaction needed to be triggered by the gravity stimulation. And then the phosphorylation correlates with the interaction.
Yuji? Actually, I had a very similar question. So how the gravity perception of the gravity would increase this interaction and would make the lazy phosphorylation, right? That's the question.
Very important. So for answering your question, we have two hypotheses. The first is the aminoplast sedimentation trigger this interaction and the phosphorylation, and the lazy come back to localize on the aminoplast. Another one is probably there's other mechanisms that can trigger these interactions. So what we have done is that we compare the phosphorylation of lazy in wild type and in PGM1's mutant background, that kind of very slow sedimentation. We found that the phosphorylation increase is exactly the same. So that means that there are unknown triggering mechanism.
Yeah, that would be my question. So basically you think this happens independently of the statolith sedimentation?
Yeah, so we think so. That's why.
That's very cool.
Yeah, but what kind of proteins or structures can sense this kind of position change? I have no idea.
Sure.
To continue to go. Yeah.
Sure, maybe as Denisa and others said, the leaving of the statoliths from the upper side might be also relevant signal. So
you said the leave?
So no, the departure of the statoliths from the upper side of the columella cell, which happens before, of course, they will sediment all the way down. Right?
But also in PGM1's mutant, the detach is also slower. Yeah.
Okay, that's a good point. Yeah. I have a second question. Okay, so your mutant screen in Fiscomitrella, right? did you find an overlap to the SGR screen from Arabidopsis, or there were all different regulators that you found?
Yeah, so the short answer is not yet, because for those random mutant, we have not characterized the genes. Okay. So what the easier one is the opposite one, because the opposite one are easier to be obtained, and we can collect multiple, and then we just to sequence there are overlap ones. Very easy to pick the gene.
Sure.
Yeah.
Thank you very much.
Thank you.
So for the fiscomitillator lazy mutant, at first, congratulations for the clear phenotype.
Thank you.
And it looks like tip growth may not be regulated by gravitropism, but position of side branch initial cells may be affected because all protonema grows in the same directions or straight. So maybe initial position may be changed. No, for the mutant, lazy mutant.
Oh, for the lazy mutant. Okay.
Yeah, this one.
Yeah. So for this, the wild type Phascometrium, they're growing upwards, the proteinema. But in this mutant, you put here, they grow toward all different directions. If you change the orientation, they will continue to growing. There's no.
So they do not bend.
Yeah, they do not bend. There's no this kind of gravitropic response.
So it means at first,
no this response.
New stem cell is regenerated as a side branch. So when you put the protein matter, and new side branch initial stem cells are formed, at that time, direction of the growth may be regulated by lazy rather than the later growth direction.
Okay, so actually we check at the very early stage. So for this tip cell, there's even no kind of proliferation. It's just
so the initiation of the side branch, side protrusion. At that time, first nucleus moves to the positions. So maybe it may be related to the nucleus movement.
Okay.
For the positioning.
Okay.
So it will be interesting to observe.
Yeah, actually, I wanted to point to this mutant, this kinesin mutant. So we identifying the single genes mutation reverse the phenotype. Actually, the family, there's 4 kinesin, and this kinesins can move nucleus. Knock out these 4 together, the nucleus will accumulate on the tip. That's a result from, I think, a professor in Nikoya's university.
Does MPK3 or MKK4 express in columella cell, or somebody showed that their expression is together with lazy protein?
So your question is that the localization, right?
Yeah, where they express and they're localized.
The MAP kinase, the localization is the distribution broadly is kind of within the cytoplasm, and could be some of them to the plasma membrane.
Yeah, but in your case, whether they express together?
There's a co-localization, some overlap, but not all the same. Because as I mentioned, the MAP kinase is expressed in all different kind of tissues, include the columella. And within the cells, the MAP kinase is distributed in the cytosol. That's possible to interact with the membrane protein.
So in the model, how they behave? Like what is the signal? And then MAP kinase perceive, and then it changes oxygen flow or something else?
Yeah, the upstream signal, we don't know. As you raised the question, we want to know, but we don't know yet.
Thank you very much.
Yeah, thank you.
Last questions. Thank you very much. Great talk and work. I have 2 very short questions. One is related to that of Professor Schweigheimer's. So you said that MAP kinases phosphorylate lases, and that there's more interaction of MAP kinases with lases upon gravity stimulation. But doesn't your data show that you actually need MAP kinases for localization of lazy to chloroplasts even before gravity stimulation? That's my first question.
That's a good question. Yeah. So usually when you grow a plant, it's very difficult to make exactly downstream growth. So it means that the plant always is kind of dynamically responds to the growth conditions, even on the medium, right? So they grow in meet some particles, they will change. So that's one explanation. Another one is that when you collect the material to do the experiment, the direction always change. So there's some start localization. But during stimulation, the interaction accumulation increase. That's the possible explanation.
Okay. Okay, maybe we can discuss this a little later in person. The second question is just...
And also, probably there are some other interactions beyond what I proposed. For example, there are also phospholipid on the amino plus surface. There are some interactions.
My question was just whether you require MAP kinases for amylo plus localization of lazy. That's all. Even before gravity stimulation. But maybe we can discuss that in person. The second question is, have you looked at, I don't know, some microtubule end markers in protonema? I mean, whether they are polarized in any way, whether the microtubule filaments are polarized in any way in growing protonema?
Yeah, very nice question. Yeah, so for this, we actually, during this kind of screens, we identify multiple of this kind of kinase for this allele. And also this one is an adapter. Then we use the adapter protein to do the turbo ID analysis for the interaction proteins. We found some catenin proteins was involved in to be the possible cargoes. So we think the cytoskeletons, the new generated cytoskeleton and their directions are important for trigger the tropism.
Thank you very much.
Thank you.
Okay, thank you, Haodong.
Okay, thank you very much.